Hi, I'm Michael Hicktel. I was diagnosed in 2017 with stage 4 lung cancer. I'm treated here by the PA, but I also do a lot of advocacy work, and I'm here supporting the team here at PA in their research endeavours and excited about looking at the future.
I'm Professor Ken O'Byrne. I've had the privilege of looking after Michael for the past 4 years now. And I think Michael is a really good example of the importance of research. Basically, Michael has an unusual mutation in a gene in his cancer called an EGFR mutation. And that was only discovered by really state-of-the-art research and then subsequent to targeting of that mutation with a small molecule called an EGFR tyrosine kinase inhibitor. Now, specifically at the moment, Michael's on a drug called osimertinib, which has only come through clinical trials in the relatively recent past. And because of that, he has benefited in terms of the tumour shrinking, his disease being controlled, and leading what I think is a relatively normal life, except for the times he comes to see me, when I think I do induce a bit of anxiety with regards to his test results and the scan results. But he's a classic example, though, of how research has really made an impact on lung cancer. Before this, people like Michael would have had a survival of 12 to 15 months. That's about the average survival for his disease. He's still alive and well. His disease is controlled after 4 years, and we're hoping we'll get 4 to 10 or 12 or 15 or 20 more years. It just depends how things go.
That's right.
Now, the key is that all of these tyrosine kinase inhibitors eventually become resistant to therapy. Michael's very aware of this. And research is now developing new drugs that are coming through that are having an impact in the resistant setting, that are actually lengthening life even further. So I think the reality is that we can't do any of this work or contribute to the global effort for this without getting funding for research. Research is the absolute key. And the work that PA Research Foundation do in raising funds and supporting our efforts are really cannot be overestimated or overstated.
Hello everyone, my name is Arthur Kulasinga and I'm the Spatial Biology Group Leader at the University of Queensland Diamantina Institute. My lab tries to understand the tumour immune cell interactions in lung cancer, and we try to identify predictive biomarkers of response to checkpoint inhibitor therapy. We've been able to map all the cells within patients' tumors, including Michael's, and that's now giving us deep insights into the interactions of the tumor cells, but also the tumor immune cells within those biopsy samples. So we're able to look at about a couple hundred proteins and all the genes that are being expressed on those tissue samples. So the material we work with, like Michael's, enables us to do the foundational research, which then can set up a clinical trial to recruit patients similar to Michael, where we're able to then inform clinicians on the findings in that research-based setting. So that's enabling that next generation of research and being able to interact with the clinicians on that work. So having the PA hospital co-located with the Translational Research Institute enables us to walk across, collect patients' tumors and biopsy material, blood samples, bring them over. And it's often warm when we bring those. So, I always tell my students that, these are precious samples, and we work from, the consenting of patients all the way through to the experimental analysis and the reporting. So it's that full gambit of translational work. So the work's now identified a DNA signature that we can predict which patients are likely to respond to second-line immunotherapy, and we're trying to test that now in a first-line setting. So it's really, really exciting. It's cutting edge. We're grateful for the support from the PA Research Foundation. The work's really enabled us to do a deep dive on the tumor microenvironment, and we've identified a stromal signature of response to second-line immunotherapy in lung cancer. The work is we're trying to now validate that signature in independent cohorts as well as first-line immunotherapy treated cohorts too.